Hi, everyone. I'm super happy to be here at this wonderful initiative, which is the Plant Genomes Initiative. And today I will be talking about this chromosome-resolved assembly that we obtained for Darwin's giant daisy trees, the Scalesia tratiloides, and a bit with a relationship to the plant island syndrome. Before I start, I'm doing all the talking, but I would like to acknowledge all the work and help I've had from wonderful set of collaborators, which I can't just read all their name without going over time. But I'm doing all the talking, but these people have really helped me process and analyze the data. When we talk about biological success, we usually talk about species richness here in green, the ecological variation, or the phenotypic diversity that a species or a group of species has. And particularly when we talk about phenotypic diversity, we talk about those monsters that look very different. And what we observe on islands is just this pronounced evolution of some phenotypic traits. And this is what we call the island syndrome, these extreme or relatively extreme traits that evolve repeatedly on lineages on different archipelagos. And so you see there, I have an animal example here, but we have the opuntias from the Galapagos here in the middle that have like these crazy trees, and we have dragon blood trees in Socotra that just look kind of alien. And so this is what I will be referring to when I talk about the island syndrome, is just the repeated and relatively extreme evolution of phenotypes. Another aspect about the island syndrome is that with the particular conditions that we have on islands is that we expect differences in terms of defense because the herbivore pressures will be different on islands or on continents, differences in dispersal, because if a plant throws all its seeds to the water, it's not going to survive. Reproductive differences are also expected, and size changes, with many organisms evolving to become trees on archipelagos, and also very diverse leaf morphologies. What we know from the Scalesia radiation is mostly due to this wonderful paper by Mario Fernandez Mazuecos. We know that the colonization of the Galapagos by an ancestral lineage was about 1 to 3 million years ago. And we know that there are 15 species now in the Galapagos, possibly some more, that just occupy a lot of different ecological niches and have a lot of these characteristics from the island syndrome. As you see, I have these leaf silhouettes here, and you see all these extreme variation in terms of leaf morphology. And we also have other traits like, for example, the fast growth, the growth to tree habits, as I was talking about. And on the genomic level, we could say that some expectations or from these insular plants is that we verify that it's a paleopolyploid, and it has the capacity for hybridizing. So we were interested in whether there was a genomic basis to all these phenotypic changes that we observe in Scalesia, and what can we observe and learn from Scalesia in the context of island syndromes. This work is already on bioRxiv. I'm currently working on the reviewers' comments, which have really substantially helped us improve the manuscript. And we begin on the assembly. We did a combination of long reads with PacBio long reads, high quality, with scaffolding with Hi-C, and then we used PacBio IsoSeq, so long read transcriptomes to annotate the genome, and we ended up with a 3.2 gigs assembly with 43,000 genes, and 76% of the content is just repeats, and I think 50% of it is long terminal repeat transposable elements, which is crazy, but this is within the ballpark of what we know from Mastracey. One thing that we noticed, and if you are familiar with BUSCOs, it's this conserved group of genes that we use to assess the quality of an assembly. And you do see that there's a low fraction of fragmented or missing conserved genes that someone curated for us. But then the really cool thing here is just this blue part, which is we see a lot of duplicated genes that are still there from the paleopolyploidization event. And so from all these complete genes, a lot of them seem to be on duplicate level, duplicated. And this was when our collaborator Lauren told us, you really have to separate the true subgenomes or the true ancestral species that exist in this assembly. And of course, this was a lot of pain to get done, but just stepping up back a bit to the polyploidy, we have ancestral species A in turquoise and B, and then they join together and form this allopolyploid species that's going to be turquoise and yellow. And if we represent it on a phylogeny, we have this. We have the separation, a speciation event generating species A and species B, and then they join together on the polyploidization event, leading to what we know here, Scalesia. It was a lot of pain to get this done, but what really worked well for us was to focus on conserved genes, so on conserved ortholog sets, and then using fossil transposable elements. And for the first step, what I did was like, I mapped these conserved genes and I asked which genes are in duplicated levels. And then I did this pairwise matrix exploring the relationships of conserved genes and their chromosomes. And you see that, for example, chromosome 1 and this 16, they had 14 genes in common. So I consider that a pair. At this point, I don't know which member is turquoise, which member is yellow, but I know that these two are a pair. And by doing this on the full scale, you see that I could see number 10 is pairing with 116. So I could get this nearly perfect one-to-one correspondence with some examples of more of chromosome pairs with less synteny. But I could get this done, and it was really good. To actually then dissect which member of the pairs was yellow and turquoise, I relied on transposable elements, and I decomposed the genome on k-mers, which are just this composition of the genomic sequences on an arbitrary number. So k-mer of 3 is represented here in blue and red. And basically, I showed you this. The idea behind separating the subgenomes based on transposable elements is just like this. We have an ancestral species which has a speciation event and generates A and B, and then we have the allopolyploidization event. And what happens is that the genomic viruses, the transposable elements that are active in this period are going to be different, or they will all be different. And so we would expect the species to have, here you see species A to have transposable elements active in this period. They would be the yellow ones, and then the green ones would be for the B. And so I was interested in just targeting yellow and greens and then separating them. How did I do this? This was done by doing k-mers of 13. And then I said, I want k-mers that are present at least 100 times at each chromosome, because I want to actually target repeats, so repeated areas on the genome. And then I want any given k-mer to be twice represented in either member of the pair. So for example, if one pair was 100, the other one had to be 200 or more. If one pair was 500, the other one had to be either 250 or less, or more than 1000. And using this information, I could separate the two subgenomes, and this really helped us and then obtain those two ancestral genomes that are inside the assembly of Scalesia. To actually date their separation, I did some node calibration with Helianthus, and this was confirmed now with KS plots, and I obtained an estimation of 4 million years. So it comes across, if we use Mario's date, that they became polyploid before coming to the Galapagos. And then the polyploidization event, what I did was resorting again to the transposable elements, but now focusing on the recent ones, on the transposable elements that are equally and evenly represented on both subgenomes, which would be here the principle for red. And then what I did was like, I want transposable elements also present in Helianthus, so then I can do genetic distances. So basically, you see, this is from a paper that I read and I got inspired. And here in red and blue, I'm looking at the figure to the left. We have transposable elements on subgenome A and B, red and blue in this case. And then the black ones would be from the outgroup. And then we calculate the pairwise genetic distances between A and B, and we calculate the pairwise genetic distances between A and B and the outgroup to estimate possibly when they joined. This opened up a new world for us, because then we could actually quantify isoforms in subgenome A in blue and in orange subgenome B, and you see that there are more isoforms there are more genes and more pseudogenes on A. So A seems to be retaining more genes, but also pseudogenes for some reason than B. But then the length of the transposable elements per chromosome is actually quite similar. We could do this chromosome stability plots, and we did see, so above you have subgenome A, below you have subgenome B, and you do see that, for example, 13 and 15 are conserved, the same with 18 and 8, but then there are some of these that kind of move around, and this was something cool for us to observe, that some of these chromosomes were actually conserved and others not. To answer the original question of, is there a genomic basis to the plant syndromes, we did the NDS ratios, and we found 920 genes under selection, which corresponded to some of the things I have been talking about. A lot of leaf genes seem to be there, and also vegetative growth genes, but also we found some osmotic salt stress genes, which could correspond to the evolution to the arid landscape of the Galapagos. And this is the image on the paper, which is a bit kind of unorganized, doesn't say much. But when we look at the Arabidopsis literature, we find that some of these genes that are the orthologs in Arabidopsis, so the genes that we find in selection in Scalesia, its orthologs on Arabidopsis make it grow bigger. They delay flowering, so the plant can be a bit longer on vegetative growth, which would be these genes here. And then genes that also regulate the transition between embryo and vegetative growth. One last thing that I looked at was at genomic expansions at gene family level. And so groups that duplicate the genes. And here we observe a similar pattern as in the selection genes, with, for example, some genes with response to water deprivation, genes associated with development of structures, so regulation of organ growth, regulation of leaf development. This group here also has regulation of xylem system development. We also see some water and hormone regulation here. And so we do see coherent signatures of natural selection, but also gene family expansion. Now, before finishing, this is a Research Council of Norway project, and I was talking about the genome assembly and annotation, but we also have some transcriptomics, transposable element dynamics coming, and some population genetics and synthesis also coming. Thank you for your time.